In this first lecture of the lecture series on dermatology, we'll be talking about the basics of dermatology, that is the structure of skin. It is important to know the various layers, the cells, and the various proteins in the skin to understand the various inflammatory disorders and vesicular bullous diseases. So let's start with the structure of skin. Skin is a stratified structure. Okay? Stratified means layered. So it is three layered: epidermis, dermis, and the subcutis. The epidermis and the dermis are separated by dermoepidermal junction. So if you see in this picture, you will see this layer separates the dermis from the epidermis. So the layer above this is called epidermis, and below it is called the dermis. The bottommost layer is the subcutis. And this layer which separates the epidermis from the dermis is the epidermal junction. In the dermis, you see various appendageal structures and the cutaneous receptors. Okay. This gland is the sebaceous gland. It is attached to the hair follicle, and another structure attached to the hair follicle is the apocrine gland, which opens along the hair follicle, and there's one more gland called eccrine gland, which is a sweat gland and which is opening directly onto the surface of skin. Okay, in addition, you see various cutaneous receptors in the dermis. Okay, so the structures include, first structure is hair follicle, sebaceous gland, apocrine gland, and eccrine gland. In addition, you see various cutaneous receptors. These are the structures embedded in the dermis. Okay? And if you see this skin under the microscope, and if you stain it with eosine nematoxylin, you would see the basement membrane, which separates the epidermis from the dermis. So the uppermost layer is epidermis, and below the dermal epidermal junction, you will see the dermis. And this layer is the dermal epidermal junction or the basement membrane. As you can see from this picture, you are seeing some downward projections from the epidermis and there's a certain upward projections from the dermis. The downward projections from the epidermis are called the rete ridges. You can see this is a downward projection, and this is the downward projection of the epidermis. This is the downward projection of the epidermis. And you can also see the upward projections of the dermis. Okay, this area is called the dermal papilla, or the papillary part of the dermis. Okay, so this layer is the epidermis, and this is the dermis. The upper projections of the dermis are the dermal papilla, the papillary dermis, and the down projection of the epidermis is called rete ridges. In the dermis, you see two layers. First is the papillary dermis, which we saw now, the upper projections of the dermis into the epidermis. And second layer is the reticular dermis. Okay. And what do these dermis contain? The dermis contain the appendageal structures embedded in it. So what does dermis is made up of? Dermis is made up of three things: cells such as fibroblasts and immune cells. The immune cells include lymphocytes, macrophages, neutrophils, eosinophils, and mast cells. And the fibroblasts is an important cell which secrete the fibers, to synthesize the fibers and secrete the ground substance. The fibers include the collagen and the elastin are the two important fibers in the dermis. The ground substance include hyaluronic acid, heparan sulfate, and the dermatan sulfate. So dermis is made up of two layers, papillary dermis and reticular dermis. And the dermis is made up of three important things. First is cells, second is fibers, and third is ground substance. Cells include fibroblasts and immune cells. And the fibroblasts synthesize the fibers and the ground substance. The fibers include collagen and elastin. The ground substance include hyaluronic acid, heparan sulfate, and dermatan sulfate. Okay, this is about the dermis. So let's understand the structure of epidermis. The basement membrane, and immediately above the basement membrane there is a single layer of cells. Okay, this layer is called stratum basale. Okay, so what it is made up of? This stratum basale is made up of cuboidal or columnar shaped keratinocytes. Keratinocytes means single layer. And along with these keratinocytes, you see two other cells. Okay, they are immigrant cells. These cells are derived from the other structures. Skin is derived from ectoderm. Okay, whereas there are certain structures which develop from other sites and they migrate during the development process. And the cells which are seen in the basal layer include melanocytes and Merkel cells. And above the stratum basale, we talked about stratum spinosum, which is multi-layered structure. It will be around 3 to 8 layers. So this structure is the stratum spinosum. So this is also made up of keratinocytes. It has multiple layers, around 3 to 8 layers. And in this layer also you will see an immigrant cell called the Langerhans cell. Immediately above the stratum spinosum, you see the layer which contains granules. It's called stratum granulosum. You can see this is the cell, and within the cell you see certain granules. That's what's called stratum granulosum. And this layer is around 2 to 3 layers, and this also contains keratinocytes, and the keratinocytes contain the keratohyalin granules and outline bodies. Okay, keratohyalin granules plus outline bodies or lamellar bodies. These keratohyalin granules are made up of profilagrin, loricrin, and involucrin proteins. And the outline bodies or lamellar bodies contain lipid vacuoles. And this layer above the stratum granulosum is a nucleated layer. You don't see the nucleus in it. Okay, you can see nucleus in the basal layer, in the spinous layer, and even the granule layer. But in stratum corneum, you don't see any nucleus. It's called stratum corneum. Okay, it's a anucleated layer. So if you see the cells between the keratinocytes in the stratum spinous layer, you would see these spines. Okay, this is a keratinocyte and another keratinocyte. In between these keratinocytes, you see these spines. This spine are called the desmosomes. Okay, these bind the adjacent keratinocytes to each other. So what happens in the epidermis is keratinization. Okay, so the cells migrate from the basal layer to the corneal layer. Okay, this process of migration of cells from the basal layer to the topmost layer, that is stratum corneum, with associated cell changes is called keratinization. So what happens in keratinization? So in the basal layer, the cells proliferate, the keratinocytes proliferate. At any point of time, at least there'll be 30% of the keratinocytes in proliferation stage. So once they proliferate, these cells migrate upwards into the stratum spinosum. What happens in the stratum spinosum? The cells undergo differentiation. Okay. And when they migrate along the upper layers of the stratum spinosum, they acquire certain granules. Okay. And they migrate onto the stratum granulosum layer. That is why it's stratum granulosum. The granules include the keratohyalin granules and adrenaline bodies. Keratohyalin granules contain profilagrin, loricrin, and involucrin. So, what is the role of the profilagrin? Profilagrin. So, filagrin, filagrin. Fil means filament, and agrine means aggregate. So, it aggregates the filaments. So, it aggregates the keratin filaments. And along with this other two proteins, loricrin and involucrin, it forms a proteinaceous structure. Okay. And this proteinaceous structure, along with the lipid found in the outline bodies of the lamellar bodies, they form the lipoproteinaceous structure, the stratum corneum. Okay, this lipoproteinaceous structure is the barrier of the skin. Okay, so we need this barrier. That is why this cells undergo change in the morphology, and they undergo certain changes, acquiring granules, and changing the size, and changing the shape, and forming the structure. Okay. So this process is called keratinization, and it usually takes 28 to 40 days. Okay. This is called epidermal turnover time. Epidermal turnover time. It's generally 28 to 40 days. But in case of psoriasis, well, there will be more inflammation, and because of the presence of inflammatory cytokines, there will be hyperproliferation of the keratinocytes. Okay, the cells will proliferate faster and they'll migrate faster. So this total time is reduced to 10 to 18 days. Okay, in psoriasis. So what happens in keratinization? Cells become flat, cells enlarge in size, cells lose their nucleus, and the metabolism is stopped. Okay, this is the final thing which is happening in the keratinization. So what happens in the keratinization is the cells migrate from proliferate in the basal layer, and they migrate towards the spinous layer, and they in spinous layer they undergo differentiation and they acquire certain granules, and they migrate to the granular layer, and in the granular layer they form the lipoproteinaceous structure which moves towards the stratum corneum, and this stratum corneum is a defense barrier. Okay, and it prevents the loss of water, and prevents the entry of the microorganisms, and it prevents against the chemical and physical injuries. Okay. And finally, what happens is the keratinocytes become flat, they become uh, anucleated, that is, they lose their nucleus. Okay, then the metabolism is stopped. You can see from this picture there is complete loss of nucleus. The cells have become flat, they have enlarged in size. Okay, and the metabolism is stopped. Okay, and in psoriasis, what happens is there is hyperproliferation of the keratinocytes. So the duration is reduced to 10 days. Okay, so they do not undergo all this transformation. So sometimes they retain the nucleus in the stratum corneum. And suppose if the cells retain the nucleus in the stratum corneum, it's called parakeratosis. The parakeratosis is retaining nuclei in stratum corneum. And this stratum corneum has to be shed. Okay, this stratum corneum has to be shed with the help of proteases and sulfatases. Okay, has to be shed. If it does not shed, what happens? There will be accumulation of the stratum corneum, which will happen in psoriasis, and it is called hyperkeratosis, which is multi-layered stratum corneum because of improper shedding. Okay, this is about the structure of epidermis and the keratinization process. So let's understand the different types of cells in the epidermis. We talked about four types of cells in epidermis. First is the keratinocyte, okay, which is the most important cell, and the other two cells, immigrant cells in the basal layer, which are called melanocytes and Merkel cells. And we talked about another immigrant cell in the stratum spinosum, which is called Langerhans cells. So we'll talk about these cells. So first is keratinocytes. So keratinocytes are the most important cell. They form more than 90 to 95% of the epidermis, and they are derived from the ectoderm, and they are non-dendritic cells. And these keratinocytes contain keratin. This keratin is divided into two types: type 1 keratin, type 2 keratin. And type 1 keratin are also called as acidic keratin, and type 2 is called basic keratin. And these keratins are numbered, and type 1 keratins are numbered from K9 to K20, and type 2 keratins are numbered from K1 to K8. Okay, these two keratins, the type 1 and type 2 keratins, they dimerize. Okay, they form a dimer, heterodimer. So keratin, type 1 keratin always pairs with type 2 keratin to form the heterodimer. So they form heterodimer. And in the basal layer, where you will see columnar shaped keratinocytes, you see the keratins 5, K5 and K14. K5 is a type 2 keratin and K14 is a type 1 keratin. So they form the heterodimer. And the cells move from the basal layer to the suprabasal layer. The differentiation, the expression of keratin 5 and 14 is reduced, and the expression of keratin 1 and keratin 10, okay, this is increased. Okay, type 1, K1 is a type 2 keratin and K10 is a type 1 keratin. An hyperproliferative condition like psoriasis, there is different expression of keratin, which is K6 and K16. Okay, this is important. And the next important cell is the melanocytes. So where do the melanocytes situated? So they are situated in the basal layer, and these are immigrant cells. So they are derived from the neural crests, and they migrate to the skin during development process. And these cells are dendritic cells. Dendritic means these cells are thrown into, or the cell membrane is thrown into folds. Okay, the melanocytes contain two melanosomes. Okay, melanosomes are eumelanosomes and pheomelanosomes. This melanocytes gives color to the skin. Okay, and these two organelles are the organelles situated within the melanocyte, which gives the color. So eumelanosome contains eumelanin, which gives the black color. Okay, and melanosome contains pheomelanin, which gives the red, brown, or to yellow color. Okay, so based on the concentration of these two melanin, eumelanin, pheomelanin, we get the constitutional color of the skin. And eumelanosome is a spherical shaped structure, and pheomelanosome is elliptical shaped structure. And after production of these melanosomes, the melanocytes, these are transported to the keratinocytes through the dendrites. Okay, that is why the melanocyte is a dendritic cell. Through the dendrites, the melanosomes are transferred to the keratinocytes, and one melanocyte transfers the melanosomes to 36 keratinocytes. That means one melanocyte interacts with 36 keratinocytes. So the ratio is 1 is to 36, and this is called epidermal melanin unit. Okay, and third cell is Merkel cells. So Merkel cells are also seen in the basal layer. They are also Immigrant cells, they are also derived from neural crest. Okay, they are non-dendritic cells and they are cutaneous receptors. Okay, they are cutaneous receptors. So these are receptors for touch and they are slowly adapting. And the fourth cell is the Langerhans cell, which is in the stratum spinosum. It's an immune cell and it's a dendritic cell. Dendritic cell. It's an immune cell. So it is derived from the bone marrow or the mesenchyme. Okay. So what is the role played by Langerhans cells? They process the antigens which enter the skin. So suppose the microbes enter the skin, these are the first cells to act against the microbes or even any other chemicals. Okay, if any insult occurs to the skin, the first cell to react is will be the Langerhans cells. These are the antigen presenting cells. Once they process the antigen, they move towards the lymph nodes and they activate the lymphatic system. Okay, so these are the four cells in the epidermis: the keratinocyte, melanocytes, Merkel cells, and the Langerhans cells. It is easy to remember the various layers of epidermis with the mnemonic boys say girls look cute. So B for basal, S for spinosum, G for granulosum, L for lucidum, and C for corneum. Okay.